You've been looking at the treatment of high-risk, low-grade glioma. What was the challenge that you were particularly wanting to illuminate?
Yes, we started this trial a long time ago, and I've presented the data from these first result trials 2 years ago here at ASCO. Actually, we included patients with a high-risk glioma, meaning patients in a need of a treatment having some predefined symptoms like age above 40, residual tumor, uncontrollable epileptic seizures. So this group was then randomized in an international trial, in a multi-institutional trial conducted by the EOITC together with the UK and together with Australia, with the NCIC Canada. And we randomized between radiotherapy alone and temozolomide alone. And the results showed that progression-free survival, this was equal. Both treatments were equal. And in addition, this trial, what was new, had a prospective question on molecular markers. So prospectively for all patients, tumor tissue was collected, and we did in parallel the molecular markers.
Right, tell me about these markers, because there are 3 of them, aren't there?
Yeah. So the first one, which we also had as main endpoint, was 1p19q deletion. And this is one of the markers. And now what we did during the last 2 years, additionally, we looked at the IDH1 and 2 mutation and also at the MGMT promoter methylation.
As a result of looking at molecular markers, you've been able to report on a slightly alternative outcomes in a subgroup, haven't you? What did you find?
Well, that was interesting actually, because we found now a predictive marker. So we found actually we subgrouped patients according to their markers. We just not looked at one marker, but we found subgroups. So let me allude. The first one is the patients who have wild type IDH1/2. That means they are not mutated, and those patients do worse. Their median survival in this was only 19 months. That goes, if I can say so, to higher grade glioma. They have a similar median survival. Then we had 2 other subgroups which are grouped by the IDH1 mutated, and these can be subdivided into the 1p19q deleted and the 1p19q non-co-deleted. So if they have both mutations, IDH1 plus and 1p19q, these are the best ones. They can live up to several years independent of the treatment.
And looking at whether they had radiation or temozolomide, what was the difference between the overall result?
For the other 2 subgroups, there was no difference for the good ones. They had either radiotherapy and temozolomide for both mutations, a good and long survival. And for the temozolomide arm alone, for the non-co-deleted ones, we have not enough events yet to have a conclusion on that one.
So what is coming out of this clinically then?
Clinically, that would mean that those 3 subgroups would have to have different treatments. And this is now under a big discussion. What should we propose now? Yes, for the worst group, at least they need radiotherapy. That could be radiotherapy alone, as my data show that could be enough, and not temozolomide. But maybe we can also say we treat them like the glioblastomas with a different treatment scheme, with radiotherapy plus chemotherapy at the same time.
So what should cancer doctors read from these data at this point?
For the bad group, for the worst group, the IDH1 wild type ones, they should learn that they should not give temozolomide in first instance, but radiotherapy. And then as a second, you can think over whether you should add something on a temozolomide or not. Yeah, whether you should give a little bit more aggressive treatment. And for the other subgroups, there may be one is enough. Yeah? For the good prognosis one, maybe it's enough to give just simply alone temozolomide or radiotherapy. And the third arm, which is a mixture in between, that will need, I would think, a combination of radiotherapy plus something. And there we have data from another trial we could add in here.